The second lecture in the Unit 1 stem cells is 4 essential categories of biological molecules. And before we discuss in detail the cells themselves, it's important to have an understanding of the individual molecules that make up those cells. The 4 molecules that we will cover are proteins, carbohydrates, lipids, and nucleic acids. If we look at each of the 4, we'll start out with this detail. Proteins, nucleic acids, and carbohydrates are actually more complex molecules that are made up of individual subunits. The subunits are called monomers. The more complex molecules, macromolecules, are called polymers. So several monomers linked together ultimately comprise or make up a polymer. Lipids do not fall under this category. They are not more complex molecules made of individual subunits. If we dive a little bit deeper, what you will see is the proteins, being polymers, are made up of individual subunits called amino acids, and we will discuss each of these in greater detail. The nucleic acids, such as DNA or RNA, are made up of nucleotides, and the carbohydrates are made up or are comprised of monosaccharides. So first, let's start out with the proteins. What do proteins do in the cell? Well, proteins are essential parts of organisms and participate in virtually every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to cellular metabolism. Proteins also have structural or mechanical functions, such as actin and myosin in muscle, and the proteins in the cytoskeleton, which form a system of scaffolding that maintains the cell shape. Other proteins are important in cell signaling, immune responses, cell adhesion, and the cell cycle, which ultimately results in reproduction of the cell. Proteins are also necessary in animals' diets, since animals cannot synthesize all the amino acids they need, and they must obtain essential amino acids from food. Through the process of digestion, animals break down ingested protein into free amino acids that then can be used in metabolism to build some of the essential proteins needed for cellular metabolism. So what is a protein? Again, if we say these are macromolecules, they're composed of combinations of 20 different types of amino acids that are bound together with peptide bonds. And if we take a closer look at a particular amino acid, you see these 3 functional groups, or 3 groups, I should say, that comprise up an amino acid. There's an amino group here. You see it with a nitrogen attached to 2 hydrogen atoms. There's the R group, and this is the side chain that differs for each amino acid. And then there's also a carboxyl group, which is common for all amino acids. So the 2 common features that you will see are these structural regions here, the amino group and the carboxyl group. What changes each time for each one of the 20 amino acids is this R group highlighted in red in the center. Again, the amino acids are the monomers. They combine together in different ways to ultimately form the polymers, which are the proteins. There are 20 different amino acids. We can basically classify them by their different side chains. In the orange ones, this would be what is filled into the R region of the structure I just showed you. The orange ones tend to be more nonpolar and neutral amino acids. The green ones here tend to be more polar and neutral. The purple amino acids are acidic and polar, and the light blue are basic and polar. You do not need to memorize the amino acids and their different polarities. I put this up here, though, to demonstrate the differences amongst the amino acids and how you can combine them to ultimately make different protein structures. And how the different amino acids are selected we'll go into when we get to that region of the lectures. But the point here being that the amino acids do have different biological properties, which can ultimately affect how the protein functions in the cell. Now, how are the amino acids linked together? If we have 20 different amino acids, how do we get the individual monomers, one monomer here and one monomer here, to combine together to begin to form the polymer? And so what you see here is for two amino acids to form together, there's a peptide bond that is formed. This is known as a dehydration synthesis reaction, because what you have is a hydroxyl group from the one amino acid binding together with a hydrogen from the second amino acid. What comes off as this reaction process is a molecule of water, and what you get is this carbon now bound to this particular nitrogen. And you see that here in this dipeptide, the peptide bond is here between this carbon, which was the formal carboxyl group, and then the amino group was here. You see now you have a bond between that carbon and that nitrogen, and the water comes off. In reverse, how do you say then, do you break these peptide bonds? Well, it's the reverse of the dehydration synthesis reaction. So rather than a water molecule being formed, instead if you insert a water molecule, you can now break down this peptide bond and have the two constitutive amino acids. And what you see then is if you just continue to do this over and over in between these carboxyl groups and these amino groups, what you can do then is link long chains of these amino acids together to ultimately form the polypeptide chain, which is what will make up the protein. And in this slide, I'm going to switch screens just for a second. There's a nice animation here that shows, if I can move this for you, that shows how that peptide bond is formed. And so what you see here are the two amino acids, and they're joined, as we said again, by a condensation or a dehydration synthesis reaction. Condensation reaction and dehydration synthesis are two terms meaning essentially the same thing. And now if we have these two amino acids, it shows here the carboxyl group of amino acid 1 will ultimately be linked to the amino group of amino acid 2. You see the water molecule coming off as a byproduct of the reaction. Again, dehydration, water being taken away. And here you see the peptide bond being formed. And there's the dipeptide. There's your peptide bond. And dipeptide, because there's two amino acids. As you increase the number of amino acids now, it's not going to be di anymore. It'll now be a polymer. And as this last slide shows, if you continue to do these dehydration synthesis reactions, you can link many amino acids together to form that long chain. Again, instead of the dipeptide, it could be a polypeptide or a polymer. 3 or more linked amino acids create the polypeptide. And notice again that the carbon backbone has nitrogen atoms positioned along in this regular pattern. So it allows you to easily pick out where the carboxyl groups are and where these amino groups are. And that way you could easily find where the individual peptide bonds are located. If we move along in the presentation now, you have an understanding of the individual components that make up the proteins. But as we did in our animation, it has the suggestion that proteins are these linear structures formed from different amino acids. And in essence, it's a little bit more complicated than that. Proteins have structure to them. There's actually 4 different levels of structure: primary, secondary, tertiary, and quaternary. And you see them indicated here. The primary structure is the simplest. It's essentially the amino acid sequence of the polypeptide chain. As we have mentioned, the primary structure is held together by these covalent or peptide bonds, which are made during that process of dehydration synthesis. Secondary structure is a little bit more complicated. It refers to the local substructure. There's 2 types of secondary structure, if you will. The first is an alpha helix, and the second is a beta sheet. And in essence, these were discovered or suggested in 1951 by Linus Pauling. These secondary structures, you see here the alpha helix being sort of a coiled structure, the beta sheet being much flatter structures, are defined by the hydrogen bonds between the main peptide groups. And so, as we had mentioned previously when I went over the polarities of the different types of R groups that make up the amino acids, the reason I did that is because those different polarities in the different R groups can change the hydrogen binding amongst the amino acids. And that's ultimately what results in these types of secondary structures, these coils in an alpha helix, or these longer, flatter structures for a beta sheet. So the amino acids, in essence, start folding back onto each other rather than staying in a long, straight line. They start to coil up or make flat sheets to have some sort of two-dimensional or three-dimensional structure. Moving on from tertiary structure, or from secondary structure, I should say, from the alpha helix and the beta sheet, you then go to tertiary structure. And this is now a three-dimensional structure of a single protein molecule. And what happens is the alpha helices and the beta sheets now start folding upon each other into a more compact globule shape. And so here you see, once you have the structures of the alpha helices and the beta sheets, they start now interacting with one another to form a more globule type shape. And then moving on from tertiary structure, you have quaternary structure. And now this is a much larger assembly of several protein molecules or polypeptide chains coming together to ultimately form the final protein structure. And this is not all uncommon in biology at all. We often see a functional protein needing several individual subunits to come together in order to give that protein its functionality. And the protein simply doesn't work unless it has all its individual parts. And so the individual parts fold up with their tertiary structure and then ultimately come together to form the final protein molecule, and that's your quaternary structure. And here you see the individual subunits, in essence, would be color-coded. There's a red one here, orange, blue, and green. The quaternary structure is stabilized by the same non-covalent interactions and disulfide bonds as the tertiary structure here. And so you have these individual subunits interacting with one another to ultimately create some sort of stable protein molecule.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. Two kinds of secondary structure units are the alpha helix and the beta pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure.
Moving on from proteins, let's discuss carbohydrates. So what is a carbohydrate? A carbohydrate is an organic compound, consists only of carbon, hydrogen, and oxygen, with a hydrogen to oxygen atom ratio of 2:1, similar to water. The term is most common in biochemistry where it is a synonym of saccharides. So sometimes you will hear carbohydrate, sometimes you will hear saccharide. Essentially, we're referring to the same thing. Carbohydrates are divided into 4 different chemical groupings: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. In general, the monosaccharides and disaccharides are smaller carbohydrates. These are often known simply as sugars. While the scientific nomenclature of carbohydrates is complex, the names of the monosaccharides and disaccharides very often end in the suffix OSE. So if you see something OSE, think sugar. For example, blood sugar is the monosaccharide glucose. Glucose has an OSE ending, indicates a sugar. Table sugar is a disaccharide. Sucrose, again, OSE ending in the term sucrose, indicates that it's a sugar. And the one you're also probably familiar with is a milk sugar, is the disaccharide lactose. So sometimes using the suffixes in words will help us understand what the meaning. And in this case, when you think OSE, think sugar. So what do carbohydrates do? Well, these polysaccharides, they serve for a very important storage of energy in the cell in the form of either starch and glycogen. You may also be familiar with their role, though, as a structural component. You may have heard of cellulose in plants, and also chitin in arthropods forms that outer cytoskeleton in the insects. The 5-carbon monosaccharide ribose is an important component of the genetic molecule known as RNA. And so carbohydrates, you'll see, are not just important for energy or structure, but also have an important role for RNA. The related deoxyribose molecule is a component of DNA, and saccharides and their derivatives may include many other important biomolecules, again, that play key roles in the immune system, fertilization, blood clotting, and development. So we see carbohydrates many different places in the cell performing important functions. As I had mentioned, the carbohydrate molecules can occur in different configurations. You have a monosaccharide. This would be, for example, glucose. There's approximately 100 different monosaccharides that exist. There are disaccharides, for example, sucrose. A disaccharide, just like we saw with a dipeptide, is in essence 2 monosaccharides linked together. And then you have a polysaccharide, which is 3 or more monosaccharides linked together. And an example of that would be cellulose. So much like we discussed how you make a peptide bond in order to join amino acids together, let's go over how you go and make a disaccharide from the individual monosaccharide components. And so here you would see an example of 2 monosaccharides. Here's glucose, here's fructose. Again, the OSE ending indicates that they're sugars. And exactly similar to what we saw in the peptide bond formation, if you undergo a dehydration synthesis reaction, now we don't have an amino group and a carboxyl group. Those are unique for the amino acids. But again, if you see, you take this hydroxyl group and the hydrogen here, you have a water molecule coming off, and now rather than a peptide bond for the disaccharide formation, we have a glycosidic bond. And you see that bond being located here in between the 2 individual monosaccharide molecules. And now what we ultimately did is we combined glucose and fructose together to ultimately form the disaccharide sucrose. Moving along, the next group of biomolecules that we'll cover are nucleic acids. There's 2 types of nucleic acids. There's DNA and there's RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. What you see here is the individual nitrogenous bases that bind to each one of those nucleic acids. If we cover that in a little bit more detail, let's look at the individual components that make up a nucleotide. There's essentially 3 different parts here to a nucleotide. You have a phosphate group, you have a sugar backbone, and then you have a particular base. So much like we saw with the peptides, where you had the R group in the structure that changed, and that would ultimately then change the composition of the amino acid, there's something a bit similar within the nucleotides. You have a core structure that contains the phosphate group and the sugar, and then you flip out the base to ultimately change the individual properties of the nucleotide. It's a little bit complicated in that you can also change out the sugar. You will see that there's a deoxyribose sugar in DNA. You see the lack of hydroxyl group here. Versus in RNA, there's a ribose sugar. And you see that the addition of the hydroxyl group in that same position. So deoxy means take away the hydroxyl group. The ribose means that the hydroxyl group is there. And so if you see, you mix together these 3 individual structural features, and down here in this image shows the actual nucleotide itself. This would be a deoxyribose, so this would be a nucleotide that would go into DNA. You see the phosphate group attached to the sugar backbone, and then you also see one of the base groups. In this case, it's cytosine bound in this position here to ultimately create this individual nucleotide. And so we could also flip out this cytosine and put in uracil or guanine or adenine, and those would make up the individual nucleotides. Now, what is important in the formation of DNA is that certain nucleotides can bind to each other. And there's some ruling there. Not all nucleotides can bind to one another. It's different from in amino acids. In amino acids, any one of the 20 amino acids could bind to another one and pair together or be linked together, I should say, and that would be fine. In terms of nucleotides, it doesn't necessarily work that way. It works along the length of the chain. You can have any combinations of nucleotides, but because DNA is double-stranded, we see that there is some base pairing on the opposite strand that must be held consistent. So, for example, what you see is you have the two different strands of DNA. And then what you have is there's the backbone, the sugar backbone in the phosphate group, and the sugar backbone in the phosphate group of the second. And what you see is that only some nucleotides can pair together. So, for example, the thymines can bind to the adenines, the guanines can bind to the cytosines. You do not see the guanines binding to the adenines. There's no GA pairing. There's always GC pairing and TA pairing. RNA is a little bit different. RNA is a single-stranded molecule. It's not double-stranded like DNA. And so you see the same structure, though, because of the nucleotides. I will make mention, though, that these are the 4 nucleotides that comprise up DNA. You have cytosine, guanine, adenine, and thymine. C, G, A, T, it's often abbreviated. But when you go over to the RNA copy of DNA, you still see 4 nucleotides, but the one that has changed now is rather than seeing thymine in RNA, there's uracil has replaced it. So we still see cytosine, guanine, and adenine, but instead of the thymine, there's now uracil. So you will never see sort of a T in a sequence of RNA. You will only see the T coming up in a sequence of DNA. If we look at some of the structural differences in DNA versus RNA, DNA forms a double helix. It has a thymine as the 4th base. Remember we said there's A's, G's, C's, and T's. And the sugar backbone is deoxyribose. In contrast, RNA tends to be single-stranded. Again, rather than thymine, there's uracil as the 4th base, and the backbone sugar is now ribose. Moving along to the last grouping now are the lipids. What are the lipids? Well, they're a broad group of naturally occurring molecules. It includes fats, waxes, steroids, vitamins, such as vitamins A, D, E, and K. And it also includes monoglycerides, diglycerides, phospholipids, amongst others. The main biological functions of lipids include energy storage. There are also structural components of cell membranes, though, and the lipids play an important role as signaling molecules. There are no polymers, as we had mentioned, in lipids, and all types of lipid molecules are insoluble in water. Think about the old mixing oil and water experiment, and so that's a result of the lipid molecules essentially being insoluble in water. There's 2 basic groups of lipids. There's the simple lipids. These are the lipids that contain only carbon, hydrogen, and oxygen. An example of these are fatty acids and triglycerides. You see them listed here at the top. But then there are also compound lipids. And the compound lipids are those that contain elements in addition to carbon, hydrogen, and oxygen. Most often what we see is a phosphate group being added. An example of the compound lipids are the phospholipids and the steroids. And this is an example of a fatty acid now. There's two different types of fatty acids. Remember, we said these simple lipids have only carbon, hydrogen, and oxygen, and you see that here. But then they can also have different structural arrangements, and you can have the saturated fatty acids, and you can have the unsaturated fatty acids. And in the saturated fatty acids, you see all of the carbon molecules here being linked to hydrogen molecules with no double bonds in the core of the structure, with the exception of the one carboxyl group here on the left-hand side. Versus in the unsaturated fatty acids, what you see is there are double bonds in between some of these carbon molecules, and not every carbon is fully bound by hydrogens or another carbon. And so two different types, how you distinguish them depends on whether these double bonds exist in the structure or not, with the exception of that carboxyl group. The triglycerides then are composed of three fatty acids bonded to a glycerol molecule. And here you see the glycerol molecule on the left, and these would be the fatty acid chains. We just talked about a fatty acid having a carboxyl group, and then you can either have single or double bonds here depending on whether it's saturated or unsaturated. And it's the thing about triglycerides is you've all probably already heard of them. It's the main constituent of vegetable oil and animal fats. They're not all terrible, though. The thing about triglycerides is they contain more than twice as much energy as carbohydrates and proteins. So, and indeed necessary for energy storage and metabolism in the body. Yet triglycerides often get a bad name because in the human body, high levels of triglycerides in the bloodstream have been linked to atherosclerosis, which is hardening of the arteries, and also then by extension, high levels of triglycerides can lead to the increased risk of heart disease and stroke. So again, though, how do you go about forming a triglyceride? Think again, dehydration synthesis. So you take your glycerol molecule, you see here coming off this carboxyl group, there's a hydroxyl group, but it will ultimately bind to this hydrogen. You will release 3 molecules of water in this instance for each of these 3 binding sites, and then you will ultimately form a triglyceride. So glycerol plus 3 fatty acids in a dehydration synthesis reaction will result in your triglyceride and the 3 water molecules coming off. Now, triglycerides containing these saturated fatty acids, remember we had said the saturated ones are the ones in which the hydrogen molecule is bound to each of the carbon molecules. There were no double bonds, if you will, between the carbons in the structure. Triglycerides containing saturated fatty acids are solid at room temperature, and these are what we think of as the fats. And this is due to that straight chain nature of the saturated fatty acids, allowing them to densely pack to form a solid. And the majority of fatty acids in animals are saturated. Now, in contrast, when you have a lot of unsaturated fatty acids coming together to form the triglyceride, what you see then is you have a liquid at room temperature. And these are often called oils then. And if you think the double bonds now in the unsaturated fatty acids prevent that close packing of the molecules. You have these kinks that result as the double bonds are forming between or are formed between the carbon molecules. And it's those kinks that then produce a liquid state for the triglycerides at room temperature. And most fatty acids in plants, in contrast, are unsaturated. Now let's take a brief look at another. Remember we said there's the simple lipids and then the complex lipids. I'm going to show this one complex lipid only because it plays such a critical role in the structure of the cell membrane. And this is a phospholipid. As you see, you have the carbon-hydrogen backbone tail or chains that are similar to what we saw when we were talking about the fatty acids. But remember, what makes it complex now is you have an addition of another group here. And in this case, you have a phosphate group indicated here in the structure, and you also have a choline group here at the top. So now we have a few more different types of atoms rather than just carbon, hydrogen, and oxygen. You see a nitrogen group up here, and you also see a phosphate group down a little bit lower. Now, phospholipids, as I had mentioned, these are the main structural components of cell membranes of living organisms. And if I go back for 1 second, let me point out what makes these phospholipids very interesting is you have these tail regions composed of the fatty acids, and these are hydrophobic tails, meaning water-hating, if you will. These are insoluble in water, these regions of the tails. But then, due to the addition of the other atoms on the head part of the phospholipid, this is a hydrophilic head. This means water-loving. And so this is a very unique molecule because the head region of it is very favorable to water, whereas the tail region of the molecule really doesn't like water at all. And what happens then is what results in is a packing of these molecules together to where you have the hydrophobic or water-fearing regions of the molecules coming together and packing into the center of the membrane, whereas the hydrophobic or hydrophilic, I should say, or water-loving heads of the phospholipids extending towards the outer regions of the membrane. So this would be maybe the outside of the cell here, where the cells are bathed in water, and these head regions of the molecules really like water, in contrast to the center, which doesn't like water. And then this would be maybe the inside of the cell here. And again, the cytoplasm, as we'll discuss in detail, is comprised mostly, or a large portion of it, of water. And so again, these molecules, the heads like water. So it takes two phospholipids to come together to form a phospholipid bilayer. And in essence, this is what comprises a large portion of your cell membrane. And it's the way that these two molecules orient with each other. It creates this hydrophobic interior, but these two hydrophilic outer surfaces that allows the membrane to form. And lastly, as we continue on, though, talking about some of the more complex lipids, I would be amiss not to mention the steroids. The steroids are molecules, as you see here, with 4 interlocking hydrocarbon rings. This would be the first time that we saw these interesting ring structures. If you ever have to look at a structure and you see these rings, it generally indicates that you're dealing with a steroid. Examples of steroids include the dietary fats. You also see here the more common ones being testosterone, cortisone, vitamin D is a steroid, as well as cholesterol.